Well, it gives me great pleasure tonight to introduce Dr. Min Ya, who is currently serving as a postdoctoral fellow at the University of Connecticut Stores, which I know is a lovely place, but I must tell you, I think I agree with Min Ya that it's not as interesting as Cambridge. Dr. Min Ya came here in 2016 to begin her graduate studies and finished early on in COVID, in the great period of COVID. She finished up in 2021, and I think that's when you started your postdoc. She is from a part of China. I'll let her give you more details if she wishes, that is famous for pandas, but also plants. And tonight she's going to speak to us about patterning the meristem, development and evolution of the floral ground plant. Dr. Min Ya.
Thank you. Okay, I'm glad that we solved the technical issue. Now it's black. That's perfect. Thank you very much for the really kind introduction. So now let's start with something that we all agree upon. Everyone likes flowers. Even if you don't consider yourself a flower person, I am sure that there have been moments when you walk by some blooming flowers, you can't help but thinking, that's really nice. Of course, flowers appeal to the aesthetics in us, and our appreciation to flowers probably can be dated back to the earliest days of our civilizations. The morphological diversity of flowers seems to be infinite, which makes them so attractive and mesmerizing. So I love flowers. I hope it's not a surprise, but it might surprise you that the aspect of flowers that I'm obsessed with is actually the earliest developmental stages in making a flower, when they don't look like flowers at all. They mostly just look like clusters of cells with some nubbings and ribbons that looks like this. There's no elaborated shape and there's no color. Some might think that this is a less glamorous aspect of flowers, but I will argue, together with many others, that few happenings in nature are as fascinating to watch as the unfolding and growth of a flower from a tiny primordial. And one of my goals today is to convince you that this statement is true. Now, think about a flower. Let's say you're thinking about something like this. So a typical flower has 4 types of floral organs, and they are arranged in very specific positions. From the outermost to the innermost, we have sepals, petals, stamens, which produce pollens that make us sneeze, and carpels, which produce seeds that we can eat. So although the final morphology of a flower can be anything they want, all flowers have the same origin story. All flowers, they all just start from a dome of cells that we call floral meristem. And within this dome of cells, there's a group of stem cells in the center, and they will produce all the organs in the flower. So from a simple dome of cells like this to a flower like that, the very first step is to have the floral meristem produce all the organs in the flower. Organs are made in spirals or whorls, which are concentric rings at the flank of the floral meristem. Once they physically emerge as those tiny bumps, they acquire their respective identity as well. Just now I mentioned that all the floral organs are arranged in very specific positions, and that's because they are produced in sequential orders, with the outer ones being made before the inner ones. So during this process of organ production, the stem cell activity inside of the floral meristem is maintained so that there is a constant supply of cells that you can make more primordia. But every flower only has a finite number of flower organs. No matter it's 4 or 4,000, it's a finite, it's a defined number. And that means the stem cell activity inside of the floral meristem will be turned off at some point, and all the cells in the center will differentiate into carpels, which is always the innermost organs. By then, all the organs are produced in the flower. They will grow, change in their shape, color, size, and eventually become the flowers that we are familiar with. So what we're looking at here, this step, is the first step in making any flower, every flower. This developmental window is very short, only concerns a few hundreds of cells, but it has profound impact on the final morphology of a flower. A certain organ inside of a flower looks like the way it is because it was initiated at a certain position at a certain time point. So this is like the master information that will influence the final morphology of a flower like a cascade. In other words, the flora meristem determines the timing, position, number, identity of all the organs in the flower, as if it has a blueprint, and many refers to this as the flora ground plan. Each of these developmental parameters are tightly controlled and tightly interconnected, as well as interconnected. And the establishment of a correct plan requires an art of coordination between all these processes, because a misinitiated organ will lead to a misshaped flower, which will ultimately impact the plant's reproductive success. For plants of the same group, they often share the same very conserved floral ground plan. And that's why almost everyone, even without a formal education in botany, can tell that those are orchids, those are lilies, and those are pansies. But among the flowering plants, we see a huge variation in the floral ground plan, resulting in variations in each of these parameters, as well as all the possible combinations between them. And this laid the foundation to the flora morphological diversity that we see in nature. So the production and the evolution of the floral ground plan is what fascinates me indefinitely. How are the different parameters controlled? How are they connected? How is the variation generated? And how do they evolve in different lineages? If we think about a flower as a dance or a symphony, although the style of the performance or the composition can be very different from piece to piece, but fundamentally, what they all have in common is that they require the movement and the instrument to occur at the right place and the right time. And I am interested in the mechanisms that enable such coordination and precision in all performances. I can give you more metaphors, but I think by now you understand what I'm obsessed with and why it is important. So when all of us get obsessed with something, we all involuntarily look for more information to understand more about it, which is why I did. And I got knowledge and information from two different worlds. On one hand, we actually have a wealth of information about the floral ground plan in many plants. And that's information we accumulated from Carlos Linnaeus, and especially after the invention of the first microscopes in the early 1800s. Now, with the support of a robust phylogeny, we actually have a very good idea of the origin and the direction of changes of floral ground plan in all the lineages. On the other hand, we also have gained so many progress in developmental genetics, thanks to intense studies in a few model systems, such as Arabidopsis thaliana in the early 1990s. Arabidopsis for plant molecular biology research is like the mice, the fruit fly for human studies. They are extremely important. And right now, we can almost put our fingers precisely on the genetic pathways that control each of those parameters in Arabidopsis, which is very incredible. However, those two fields have been historically independent from each other. On one hand, the morphological evolution has been focusing on comparisons at the deep nodes of the tree. Although we can see the origin and direction of changes of the floral ground plan, we cannot understand the underlying molecular mechanisms. On the other hand, for developmental genetic studies, although they have been focusing on a very small number of model species at the tips of the tree, although the mechanisms of many genes are well understood, but whether or not they are involved in evolutionary changes is not established at all. Well, this is not very satisfying, right? Seems like something has to be done somehow. Obviously, I'm not the first one in history that ever felt this way. In fact, a lot of researchers have tried a lot of effort over the past decades, tried to bridge these two fields together, which actually led to a birth of a new field, evo-devo, evolutionary developmental biology. people recognize that one way to bridge these two fields together is to have more model systems. But those systems has to come from different positions of the tree, and they have also to be able to be manipulated for genetic and developmental studies. So the information we get from those new models can be relevant to the deep nodes of the tree, but can also tell us molecular mechanisms through comparative genetic studies. Well, this sounds easy, but many of us can imagine it's not a trivial task at all, which is why over the past decades, we actually don't have many good new model systems for studying the regulation of flora meristem. And there is still a lot of missing pieces in our knowledge about the evolution and developmental basis of the flora ground plant. Recognizing this gap and the necessity of closing this gap, my PhD work, my postdoc work, as well as my future research program has been, is, and will be focusing on bringing these two fields together and fill in the gaps in our knowledge. Specifically, my research program starts with two charismatic model systems, and today I will tell you two little stories. And first, we'll get a little daily dose of ecologia that I want to tell you about how variations in number and timing can be generated. And then I will give you some propaganda about mimulus so that we talk about how the positioning information can be generated. Those two hashtags, you can actually search online. You will find information about them and me. So I hope by the end of these two stories, you will be convinced that my model systems, they are extremely cute, they're very charismatic, but they're also very powerful. They hold the power to help us to understand some questions that cannot be addressed by other systems right now. Okay? Let me drink some water. All right, let's jump in the first story, daily dose of ecologia. This first story concerns a developmental process called flora meristem termination. If we go back to the original slides that I showed you, the first step in making flower, I mentioned that the stem cell activity inside of the flora meristem will be turned off at some point because all the flowers have finite number of organs. Flora meristem termination is this tightly controlled developmental process to intentionally shut off the stem cell activity for the completion of organ production in a flower. Many of us know that there are many different types of meristems in plants. Compared to the one that flora meristem produce flowers, there are also meristems that produce leaves, produce roots. But flora meristem is a little special compared to the one that produce leaves and roots because for those cases, the stem cell activity inside of the meristem will be maintained indefinitely so they can produce organs continuously throughout the lifespan of a plant. But a flora meristem will always be terminated. So this developmental process is a defining feature for any flora meristem, and it's universal for all flowers. It's also a major determinant of the total organ numbers in a flower. Let's think about it. If a flora meristem terminated early, so the stem cell activity is short, fewer whorls of organs in the flower. But if its activity is maintained longer, it terminated later, more whorls of organs in the flower, and thus more organs in the flower. If we look at the evolutionary transitions of flora ground plant among all the flowering plants, although we can see an enormous amount of variation, but there are two general trends are being agreed upon by many people. One being the transition from variable to stable whorl numbers, and the other being the transition from large to small whorl numbers. And both of these critical transitions are directly controlled by the termination process. In other words, canalization towards relatively rapid flora meristem termination is a major force in shaping flowering plant evolution. We simply cannot really fully understand the evolution of flowering plants without understanding how termination process is controlled, how is variation generated, and how is this canalization established. Well, now you can ask, what do we actually know about the termination process? Let's come back to the lab rat example for plants, Arabidopsis. For Arabidopsis, we actually know quite a lot. So Arabidopsis flower is very simple. It's very small, only has 4 whorls of organs. Each organ type occupied 1 whorl. For many years, through a very series of very elegant experiments, researchers have found a molecular pathway that actually acts like a timer to ensure only 4 whorls of organs to be produced in the flower. And later, this whole molecular pathway is being proven to be conserved in tomato flowers as well, which also only have 4 whorls of organs. Although this result is super cool, but it raises some obvious questions if we put things in an evolutionary perspective, because we have a lot more flowers have way more than 4 whorls of organs, right? So is this timer mechanism conserved in other flowers? If yes, how does it produce flowers of more than 4 whorls? And if no, what functions as a timer otherwise? For this important question, we actually cannot use Arabidopsis for tomato to study it because they belong to lineages that have the whorl number super canalized. During my PhD, I realized that the lab model system in our lab, the beautiful Echologia flower, which originally developed for study petal elaboration, look at that, is also very suitable for studying the regulation of Flora meristans. So there are a number of obvious differences between Echologia and Arabidopsis. Echologia is larger, it's prettier, and it has way more organs. Here we have a cross-section of Arabidopsis flower bud before and after Flora meristem termination. We see the dome of meristem cell is gone, resulting in carpal primordia. Here we have sections of Echologia flora bud before and after Flora meristem termination. Similarly, the dome of cell is gone, resulting in carpals, but before that, way more whorls of organs have been produced. To use Echologia to study this termination process, one obvious place for me to start is to see whether the known genes in Arabidopsis perform similar functions in my flowers too, right? So during my PhD, I have conducted functional study of a number of genes by silencing their expression in my flowers. So here we have a wild type Echologia flower of the front view of it and the flora diagram. Silencing some of the genes led to a complete elimination of organ production. And for some other genes, led to an uncontrolled proliferation of organs in the flower. And this tells us that, for example, those genes here, they are indeed involved in the regulation of Flora meristem, but it tells us very little about how the termination process is controlled. Because what we want to know is not how to get from 100 to 0 or 100 to 10,000. We want to know how to get from 100 to 103 precisely, for example. And silencing some other genes, they only have partial functional conservation in my flower. And more importantly, the key genes in that timer pathway actually do not have homologs, orthologs in Echologia genome, as silencing them doesn't lead to any phenotype. All these are telling me that this timer pathway that people discovered in Arabidopsis are likely not share functional conservation in my flower. For now, I don't really have obvious candidate gene to start with. I need to be more proactive. To find the genes, to proactively find the genes, I did a few things. First, I did RNA sequencing on those tiny meristems at 4 finely dissected developmental stages. And results from this project will be a framework data for me for any future projects, because this project tells us what kind of genes are expressed at which stage and how do they change over different stages. Meanwhile, in collaboration with Dr. Stephanie Conway, we also established a live confocal imaging protocol for the flora meristem of Echologia. So here we're looking at young Echologia flora meristems under a confocal microscope. We were able to take the meristem off the plant and grow them on petri dishes over extensive amount of time and image them under the microscope every day. By doing so, we were able to track all the cells in the flora meristem over time and then quantify how do they divide, how do they expand, the position of division, the frequency, the direction, and everything. So we get all this data and we can quantify what's the cellular behavior associated with different stages of the termination process. To our knowledge, this is actually the first live confocal imaging protocol and analysis developed for a non-traditional model system. And this will also be the foundation for us in the future to understand how do the actions of genes will influence the action of cells that lead to the consequence of development. Meanwhile, there's a genetic cross happening in the lab, and we were very excited about that. So this cross is between two sister species in Aquilegia, Aquilegia brevistyla and Aquilegia canadensis, which got some perfect intermediate F1s. And we self-pollinated one of the F1s, which gave us a population of F2 flowers that looks like this. I know, very stunning. Please don't get distracted by the colors and shapes for now. I want to remind you that I'm using those flowers to study how the termination process is controlled. Well, now you can ask me, but you just spent all this time telling us that termination happens at the earliest stage of development. How do you use those flowers? Good question. And it turned out my flowers are really good to me. These two parental species, apparently, they have the same floral ground plan with one exception. They have the same number of whorls for all organs except for stamen whorl numbers, which really opened the door. Let's think about it. So the differences in the stamen whorl numbers in these cases can actually be directly translated into the differences in the timing of flora meristem termination. If a flora meristem terminated early, fewer whorls of stamens in that flower. But if the meristem stay active longer, more whorls of stamens in that flower, right? In other words, I'm using stamen whorl number as a proxy, as a quantitative trait that we can ask what is the genetic architecture underlying this variation. In other words, I'm counting stamen whorl numbers in flowers, and here's how I count it. So in all Aquilegia flower, all the organs are produced in a whorl of 5, but the petal whorls and stamen whorls, they are tightly positioned against each other, and they alternate in position for each whorl. So for every flower, there will be a few whorls of stamens that's positioned right above the petals, and then there will be a few whorls of stamens positioned in between the petals. Let's do a little practice. So here we have 3 tiny cute F2 flowers that I have. If we're gonna count the number of stamen whorls that's positioned right above the petals, how many does this one have? 3? How about this? Above petals? 3? How about this? Nice. Now let's count the stamen whorl number in between the petals. How many does this have? 3. And well, I give you the answers now. 4. So for these 3 examples we have, their total stamen whorl number will be 6, 7, and 8, respectively. So in total, I counted about 5,000 flowers in more than 400 individuals. If we look at the mean stamen whorl number per plant, we'll see a nice distribution that the parental species are clustered at the lower and the higher end, and the F2s spread across the entire range. Now, for every flower that we phenotyped, we also did whole genome sequencing to construct a genetic map. With genotype and phenotype in hand, we can actually associate them and find out which part of the genome is associated with a trait that we're interested in, which is QTO mapping. And here's how it looks like. So on the x-axis, we have the 7 chromosomes of Aquilegia. And on the y-axis, it's called profiled lot score, which is essentially an indication of likelihood. Anything above this blue line indicate a significant QTL locus associated with a trait that we're interested in. And voila, we got a lot of QTL, in fact, 1 on each chromosome. And this tells us that the trait, the genetic basis architecture underlying our trait of variation is complex, is controlled by many genes acting together. Now, with the help with the help of my RNA-seq data, I can narrow down the genomic region in the chromosome to really look at the candidate genes. Specifically, I'm looking for candidate genes that are expressed at early developmental stages, and they are differentially expressed before and after the termination process, and genes have homologs that are being shown to studied in other species that have any kinds of meristem-related functions. With these criteria in hand, I got a list of candidate genes that I'm very excited about. And I'd like to show you one tiny example today. So this little guy, little zipper 3 under Q2. What we're looking at here is a series of sections of very young flora meristems in Aquilegia. And all the cells stained brownish purple are the cells expressing our gene of interest, little zipper 3. And this is done by a technique called in situ hybridization. It tells where our gene of interest is expressed in the tissue. So first, I'd like to bring your attention to those regions which we saw strong expression of little zipper at organ boundaries. And this is an expression pattern that have been previously observed and studied in both Arabidopsis and tomato, indicating that this part of the function probably is conserved in Aquilegia as well. And then let's look at these regions. Also have a weak expression of this gene. And this region happened to overlap with the stem cell population in the flora meristem as well. And this expression pattern has been previously observed in Arabidopsis, vegetative meristem. The meristem produces leaves. So what is it doing in the flora meristem of my flower is very curious. What is more curious is we observed a strong specific expression of this gene only at the first cell layer of the flora meristem. And this expression is so specific, and no one has ever seen this. What exactly is this gene doing? What is the function of it? I really want to know. So this is just one of the examples of my candidate gene list. In fact, this is the first list of candidate genes ever, to my knowledge, that can tell us something about how the natural variation in the timing of flora meristem termination is controlled. And we're able to get this list of genes eventually is because my flowers are really cute and they hold the power, they have the natural variation, and this question simply cannot be answered by Arabidopsis, for example. And this list will also be the starting point of my future research program because we can really fully characterize all the candidate genes. What do they do? How are they expressed? What are the functions? What are the interactions between the genes? We can build a molecular network controlling to see what's controlling what. And we can even go a step further to understand the mechanisms underlying it. How are the different genes from different species associated with the differences in the termination process? What are the potential upstream regulators and what are the downstream target? Okay, here concludes my first little story. Let's take some time to drink water and breathe, and then we'll jump in. I'm gonna drink water. Everyone breathing? Ready for some propaganda? All right. So let's talk about positions in flower. This is something that we see all the time, but we just don't think about it. Because actually for most of the flowers we see in nature can be grouped into two major groups. They can either be radially symmetric or bilaterally symmetric. The former has multiple planes of symmetry, and the latter only has one, dividing the flower into two mirroring halves, left and right. So these flowers are called bilaterally symmetric. They are also named monosymmetry because one plane of symmetry. And today I will be using those two phrases interchangeably. From an evolutionary perspective, all the ancestors of all the flowering plants and of all the major lineages are predicted to produce radially symmetric flower. That means monosymmetry is a derived state. With each update of the phylogeny of the flowering plants, the estimated transition between those two states increased every single time. And now the latest estimation of the transition between those two forms is as many as 200 times. So it's very evolutionarily laid out. But this trait is also biologically significant because people think the transition to bilateral symmetry is considered as key innovation in many species-rich groups like legumes, orchids, because this specific bilateral symmetry shape enabled pollinatory interactions, increased the efficiency. Bees can grab onto the flower more effectively. From a developmental perspective, though, bilateral symmetry can be expressed and manifested in many different ways. For example, it can be changes in organ number, organ size, shape, or color. But regardless of how the flower look like in the end, the underlying principle in making one monosymmetric flower is the same. Because having one plane of symmetry like this, it means the left and the right of the flower is the same, right? It also means the upper part and the lower part of the flower followed different developmental trajectories. In other words, regardless of how different these developmental programs are, regardless at which developmental time point those differences were elaborated, or regardless of how the flower looked like in the end, this division between the upper and the lower is a prerequisite of making any monosymmetric flower, every monosymmetric flower. And this division, actually, we can physically distinguish it since the very beginning of a flower, because when the floral meristem is just produced, it's actually being made by another type of meristem that's called inflorescence meristem. So once an inflorescence meristem produces a floral meristem on the flank, by default, half of its cells will be located relatively closer to the inflorescence, right? And we'll call that the dorsal domain. And the remaining cells will be located physically further away from the inflorescence, which we call a ventral domain. And it turned out this division between the dorsal and ventral corresponding to the upper and lower division in a flower that established bilateral symmetry. Now that we know this division of upper and lower, dorsal and ventral can be distinguished since the very beginning, here comes some obvious questions that I'm interested in. How are the domains defined at the molecular level? How is this division maintained? And how is the difference between the dorsal and the ventral manifested? Well, for the last part, we actually know something about. So in the early 1990s, people are studying snapdragon a lot by studying a ventralized mutant. So snapdragon flower is typically bilateral symmetric, and it's the most pronounced in terms of petal morphology. It has 2 dorsal petals, 2 lateral petals, and 1 ventral petal. So people are studying a ventralized mutant in snapdragon and found out that it's being caused by a gene called cycloidea. So this discovery of cycloidea started a whole period of studying of cycloidea homologs in many plant taxa. And to people's surprise, what blew people's mind is that every taxa studied to date, there's a correlation between restricted cycloidea expression and bilateral symmetry. So this cycloidea gene story actually became one of the textbook examples of how the same molecular program can be co-opted, recruited again and again to build similar functions. But after 30 years of intense study of cycloidea, we still have no idea how this restricted cycloidea division is first established, precisely because we don't have answers to the first 2 questions that I answered. Similarly, although the independent repeated co-option is really cool, but are the cycloidea genes activated in the same way? Or how deep is this parallel recruitment conservation? We have no idea. So we don't have any ideas about these. It's not because people are not interested. People are super interested, but mostly it's because of technical limitations. Because for the species, we actually have all the molecular functional tools. They somehow all have radial symmetric flowers. So leading to this very awkward situation that if they don't have bilateral symmetric program to start with, how do we use them to study how such program is controlled, right? Until now, there we have Mimulus. To our knowledge, Mimulus is the only system that not only have bilateral symmetry, bilaterally symmetric flowers, but also can be easily transformed, and we can do different kind of manipulations and studies to them. And thus, I'm here to spread you with minimalist propaganda. After this many years of studying of bilateral symmetry, of course, people have some hypothesis of what controls bilateral symmetry at the early stage. And we can use minimalist to test it. And the hypothesis is auxin. Who knows about auxin here? Yes. So auxin is one of the most major hormones in plants. It literally controls every aspect of development in plants. So when people were suspecting auxin is involved, it's actually not surprising to people at all, because auxin is involved in literally everything. But the situation here is slightly more interesting, because if we apply a chemical called MPA, which inhibits auxin transport in plants for a radially symmetric flower, what we will see is failed organ initiation. There will be fewer organs in a flower because auxin is for organ initiation. But if we apply auxin to bilateral MPA to bilaterally symmetric flower, what people also see the failed organ initiation, but there's also transition from bilateral symmetry to radial symmetry. What makes this more interesting is that it seems like the application of auxin itself is also can trigger this transition. So it seems like, yes, sure, auxin is involved in establishing bilateral symmetry, but it's the dynamics of auxin rather than the absolute concentration somehow influences that. So how can we see the dynamic of auxin? We can actually look at it. Many years ago, people have developed some fluorescent reporters for Arabidopsis that reports the cells that have high auxin concentration. So here we are looking at a top view of an inflorescence meristem of Arabidopsis. All the cells expressed in red are cells experiencing high auxin concentration. The fluorescent in dark. So when we look at young meristems at the early stage, we'll see DR5 signal across the entire tiny flora meristem. But soon after that, before organ initiation, the signal in the center disappears and resolves into 4 dots on the side, marking the initiation site of sepals. And that's a typical, very simple program dynamic of DR5. First in the center, and then you resolve on the side. Now let's look at what's happening in Mimulus. So here's my very cute Mimulus flower again. It has a typical bilateral symmetry with 2 dorsal petals, 1 ventral petal that have a lot of pigmentation spot, yellow nectar guide, and hair as well, and 2 lateral petals. That's pretty much the intermediate between them. Here we have a scanning electron microscope image of a Mimulus inflorescence. That's a top view. So what we're seeing here is an inflorescence meristem in the process of making 2 leaves. 90 degrees from that, it has the opposite phyllotaxy. So 90 degrees from that, another pair of leaves, each subtending a young flora meristem, just a bump of cells. 90 degrees from that, another pair of leaves, and slightly older flora meristem. In a second, I will show you a bunch of propaganda images that corresponding to the topmost part of this SEM. So that we'll have inflorescence meristem flanked by 2 leaves, and then below it, there will be a flora meristem with its dorsal and the ventral domain division, okay? So I put the same DR5 reporter into my plants together with a plasma membrane marker and imaged hundreds of meristems with confocal microscope. What we're looking at here is the youngest flora meristem that I can physically dissect. It's about 60 micron in width. In the middle panel, we can see that although some of the cells are showing DR5, but inside of the flora meristem, there's nothing. But this situation will soon change. In a while, we'll start to see cells expressing DR5 inside of the flora meristem. First, there will be 2 dots left and right, and then there will be more cells emerging DR5 in the center. The number of cells expressing DR5 will then increase from 1 or 2 or 3 cells that we're seeing here to as many as 20 and 30 cells. After it reaches a number of cells in the center, this signal will then move to the dorsalmost part of the floral meristem, like shown here. After this movement, for the first time, we'll see some cells in the ventral part also express DR5. Now that we have cells in both dorsal and ventral expressing DR5, expressing high oxygen concentration, the number of cells expressing DR5 will then drastically increase to a degree that as if all the cells in the floral meristem are experiencing really, really high oxygen concentration, except for a band of cells in the center, dividing the cells meristem into the dorsal and the ventral part, as if this is a no man's land that no one shall cross. Only after the appearance of no man's land, the number of cells expressing DR5 will then decrease, resolving into the 5 dots and marking the initiation site of sepal primordia. And this is what we expect to see for DR5 pattern. So I only said this is what's expected for the last part, because actually many things we saw before is mind-blowing. It's very surprising, especially this movement from the center to the dorsal and how the expressing at the ventral is much later, and not to mention this no man's land. In short, the oxygen dynamic during such a short developmental window is very dynamic, and DR5 patterns has been monitored in other systems for decades. No one has seen a dynamic thing like this. Now the obvious question is, what does this mean and does it matter? To answer this question, I put the same fluorescent reporter lines into many other mutants that I have, and I can compare the patterns of DR5 in different backgrounds. For example, here I have a dorsalized flower resulting from overexpressing one of the cycloidea genes. But the DR5 pattern in this background didn't really change, suggesting that oxygen response is indeed upstream of the cycloidea-based program. But here we have another dorsalized flower that resulted from knocking down one of the genes called BOP, and the situation is slightly different. So initially, there's still nothing, and then there will be 2 dots, left and right. And then there will be 3 dots. But instead of having the 3rd dot in the center, it's always on the dorsal side. And then there will be more dots where straight lines on the peripheral region of the floral meristem. And that's it. It's pretty different, right? To understand what are the nature of these cells, I conducted live imaging to trace the development of these cells expressing DR5 in this CRISPR line. And it turned out those cells are marking the future primordia since the beginning. Something seems to be missing here, right? It seems like in this mutant background, the oxygen did one part of its job, which is marking the initiation site, but skipped another part of its job, which is patterning the dorsal and the ventral domain. And without this dynamic movement, what we get is ventralized flower. So the ventral identity could be the default for those flowers. And now for these differences, we can actually compare that it seems like DR5 as a center or the movement or the no man's land will be crucial for the cells to know their position in the floral meristem. And this will also be the starting of some many very exciting projects in the future, because we want to know how exactly do the dynamics matter, how are they associated with different cell morphologies, and how is the positional information established in the floral meristem. We can also do more sequencing to find more genes and study their functions in the floral meristem. And at some point, maybe in 10 years later, we'll have a more complete view of how do those cells can tell their position in the flower. Okay, that concludes my 2 little stories. And I hope by now you are intrigued and agree that my little flowers are indeed very powerful, can help us to answer some really important questions. And for the stories that I told you today, it's not only just for my personal interest, although I love flowers, I want to know how they're regulated. For the results and genes that we got from those projects can also help us to further improve crops and also other plant production and control their flowering in different environment, especially for fruit, for crop production. So many things we eat are products of flowers. And the genes that knowledge we got from those projects can help us further engineering better crops for everyone. And also, those can tell us more about development of multicellular organisms like us in general. For example, when I'm thinking about what are the molecular programs controlling the timing number of whorls in flowers, animal biologists are also thinking about what is a molecular timer that times the number of vertebrates in animals. And bilateral symmetry by no means only exists in plants. Just look at us. We and our plants and our pets, we're functional, we're multicellular organisms. That's because all our cells have the ability to know their position, their identity, to assess time, to know their environment. And that's why we developed into functional organisms. So hopefully the principles and mechanisms that we uncover from those programs can tell us something about development in general. But what I really want you to know, take away from today, is that next time when you see some flowers, don't forget how they are developed from those tiny clusters of cells that from complicated developmental processes that we can't even see with our naked eye. And I hope that make you appreciate flowers a little bit more than before. With that, I'd like to thank my funding sources, my labs, and my advisors. And I'm excited to say that I'm starting my own lab next year at the University of Illinois at Champaign. So stay tuned for more flower stories from the main lab. And thank you very much for your attention.
Thank you, Minya. Are there questions from the audience?
Okay.
Are any questions here?
Oh, I love the plants since I was little. Yeah, because my dad is a amateur botanist and he loves plants, and it's just very natural for me growing up loving plants. He's those people that we walk in the woods, he will say, oh, this is that, this is that, this is that. We take care of plants in the house together. But it was only in undergrad where after I finished started my master's, I realized, oh, studying plants can be a career choice. Before that, it was a hobby. Alex?
So these stencils, did they also deal with the color of the flower turning out?
No. So coloration in flowers normally happen very late in development. So the stem cells, they are responsible for organ initiations and their arrangement. After the organs initiate, it's other things that's responsible of shaping the shape the organs and color them.
So when you added the or when you promoted the auxin production, did that skip steps in all of your mutants or some of them, or was it different
for that particular mutant, they all skipped. There's no patterning going on. It's always just marking the thing, which is fascinating, I think. Termination of it, well, yes, if, for example, if we think about annuals, they will die at some point. So the stem cell activity will be terminated at some point, but it's less, it's very, I wouldn't say it's as tightly controlled as the flora meristem, because flower is a very specific structure. So you really need a lot of different layers of molecular control to ensure everything's right. But for a plant, you know, growing outside it will more depend on the environmental conditions. You will be more flexible. It can be, terminated. It can die right now, or it can die next week. It's more flexible.
Question from the chat. I'm wondering if sound has anything to do with development of seeds and morphology. I have been struck with the transverse section of ovaries at the Harvard Peabody Museum and how they resemble patterns on the, I'm not sure how to pronounce that one. Any chance sound plays a role?
Sound, if we're talking about vibration, actually, I know some of the studies that in roots that they realize they suspect that root cells can sense the tiny vibrations that water drops touches the ground, so that like they grow to the, well, that was experiments that grow in some plants, and then there's a leaky pipe dripping water, and the roots always know where the leaky pipe is, and people suspect that could be something with the vibration, or they just somehow sense the water. So I'm not sure. I don't think I answered this question, but that's an anecdote that I heard.
Just got another comment too, that we loved your presentation.
Thank you. Yes.
And do you think that auxin overproduction would lead to speciation down the line? Or sterile?
So auxin is something that's literally involved in everything. Every plant has it, but different plants regulate auxin in slightly different ways. So is there a consequence or a cause? I think it's hard to entangle, and how much role it actually plays in reproductive isolation is hard to tell, because yeah, it's hard to tell.
Well, if there are no more questions, let's give Minya another round of applause. Thank you very much.